According to WHO, 1.3 million people each year meet with road accident and suffer from severe spinal cord injuries, which results in impaired or restricted limb movement. And transcutaneous spinal cord stimulation is expected to show remarkable results in restoration of sensory motor function in limbs. Now, let's see how we can achieve this process. So, improved understanding of human nerve function and its implementation helps us in this process by identification of nerve pathways to send signals all over the body. Better understanding of nerve pathway helps us in better stimulation technique. So, let's see what my project is about. In this study, we are aiming to determine the effectiveness of cervical transcutaneous spinal cord stimulation to produce evoke responses in the limbs. In this project, I am guided by Dr. Richard Riley, Professor of Neural Engineering, and Dr. Connor Manuk from BMR Limited. In order to get the optimum evoke responses in the limbs, we need to study the origination and termination of the nerves from the cervical region to the limb region in the human body. The evoke response in the muscle will be produced by simulating the transsynaptic alpha motor neuron in the cervical region using an array of 3x3 electrode placed posteriorly on the neck, and an array of rectangular electrodes are placed anteriorly on the neck to complete the circuit. We have to target the desired nerve for better evoked response, and this response can be achieved by altering the location of cathode over the posterior neck and by activating the dorsal root nerve while minimizing the activation of ventral root. So, this defines our aim of understanding optimal factors of electrodes to be attached on cervical region of the spine. As you can see in the video, after knowing the target nerve to achieve the muscle activity, we place the EMG electrodes on patient to gather the important data. But the major challenge in this process is that patient available are of different phenotype. That is, they are from various age, sex, color, and regions, and therefore this creates a variation in anatomies, thus creating a strict need of personalized mathematical model to be built. So, therefore, we will be using high resolution T1 and T2 cervical MRI image data obtained from the patient and thus creating a personalized mathematical model, which will ultimately aid in identification of neuronal groups to simulate and activate muscle function in the limbs of patients suffering from severe spinal cord injuries. Thank you.